My research is focused mostly on exploring the plant and the soil microbiome with functions that can mitigate biotic and abiotic stresses in plants. If you think about biotic stresses, we have a lot of soil-borne pathogens, we have pests, and we have parasitic weeds, for example. Striga is a weed that requires a host to fully develop, especially at the initial stages of the development of this parasite. It causes major damage in crop production systems, specifically sub-Saharan Africa. Basically, Striga has a mechanism to sense some chemicals that are kind of released by the host plant. And by doing that, they start the germination process. We know some microbes can produce molecules like ethylene that could lead to the suicidal germination and depletion of the seed bank of Striga. That occurs basically by stimulating the germination of Striga without having a host close by. Instead of finding that, we find some volatiles that effectively can suppress the germination of Striga. That was a big aha moment because we're not expecting that. And now we came up to a point that we identify those volatiles, those molecules, we can apply them and even apply precursor molecules that activates the microbiome to produce these functions in the system. And that's a big difference between the way we see engineering microbiome by effectively activating a function in an existing microbiome. I'm using a lot of the QIAGEN, DNA, RNA extraction kits for soil and plant materials. I'm also familiarized with the QIAGEN qPCR instrument and reagents. It's very important to have standardized operating protocols across different labs, and QIAGEN provides an opportunity to really standardize those operations. I believe we have a need for sustainable intensification, which means be more sustainable in the way we produce food, and at the same time produce more food for the growing population. And microbes are the major players that we can manipulate to achieve those goals.
QIAGEN.
Sample to insight.